Many viruses move long distances through their host cells by binding to microtubule-based motor proteins. Herpes simplex virus, for example, can bind to kinesins and dynein, as well as the dynein accessory factor dynactin. Derek Walsh and colleagues at Northwestern University in Chicago, however, were interested in the herpes virus's interaction with the microtubules themselves, particularly during the early stages of infection.
So we had shown previously that the virus induces microtubule stabilization late in infection, and this seemed to be involved in egress of the virus. But when we started to look at the earlier events, we found that there wasn't any really significant induction of microtubule stabilization. And most of the virus particles that we were looking at by immunofluorescence seemed to localize to dynamic microtubules. So that kind of gave us an initial hint that maybe the virus was using these dynamic microtubule subsets.
Walsh and colleagues, led by postdoc Vladimir Jovasevic, therefore looked at EB1 and CLIP-170, two so-called plus-tip proteins that track the plus ends of dynamic microtubules.
We knocked down these plus-end tracking proteins and found that these things just totally floored the infection. We were actually pretty shocked to see, I think there's like a 5,000-fold effect on infection.
To investigate which stage of infection was inhibited in the absence of EB1 and CLIP-170, Jovasevic et al. Examined the expression of viral gene products and found that knocking down the plus-end tracking proteins blocked the expression of not only late-stage structural proteins, but also the virus's immediate early genes.
That told us that there was a block at some point before the virus can even begin expressing its genes. And then we found that it wasn't a block to the actual entry of the virus, so the virus was getting in okay. So that drove us to then actually look at virus motility.
The herpes virus initiates gene expression after it has moved along microtubules from the cell periphery to the nucleus. In the absence of EB1 or CLIP-170, however, this retrograde transport process was inhibited, even though the cell's microtubule organization appeared unchanged. Retrograde transport depends on the -n-directed motor dynein and its accessory factor dynactin, both of which can bind to herpes virus particles. When the researchers knocked down the large subunit of dynactin, the virus also failed to accumulate in the nucleus and initiate expression of its immediate early genes. However, as well as supporting dynein-dependent transport processes, dynactin can also bind to CLIP-170 and link microtubule plus ends to intracellular cargoes such as melanosomes. To investigate whether dynactin works with EB1 and CLIP-170 to capture viral particles at microtubule plus ends, or whether dynactin simply works with dynein to mediate retrograde transport, Jovasevic et al. Compared the effects of depleting dynactin to the effects of inhibiting dynein.
What we found was that when you inhibit dynein, you get this kind of twitchy motility of the virus. The virus is blocked from getting to the nucleus. It needs dynein to move, but it has this stop-start motility in the cell. But when you knock down dynactin, the effects are actually much more potent. So instead of this twitchy movement, you just see absolutely no movement at all. So that kind of clued us into dynactin having a distinct function from its established role in stimulating dynein activity.
Knocking down EB1 or CLIP-170 also blocked long-range viral movements, supporting the idea that dynactin works with these plus-end tracking proteins to capture viral particles in the cell periphery. In contrast, knocking down EB1, CLIP-170 or dynactin had little effect on the distribution and motility of other dynein-dependent cargoes such as mitochondria and lysosomes. EB1, CLIP-170 and dynactin therefore appear to act like a receptor complex for herpes virus particles, allowing the virus to bind to the plus ends of dynamic microtubules before initiating dynein-mediated retrograde transport to the host cell nucleus.
I think people kind of assume that because the virus can engage motor proteins that it can just interact with microtubules through the motor proteins, and once it binds a motor, it's just going to move. But we actually think that it's a much more regulated process, and so even if the virus can interact with a motor, it really needs to be captured by the plus end first before it can actually load onto a microtubule and start moving. We're now trying to figure out how the virus is actually targeting this complex. It seems very critical to infection, and so what we're trying to figure out is how the virus is engaging dynactin. We're also quite interested in how this might function in neurons because the virus is neurotropic.
In the meantime, however, you can learn more about how microtubule plus end-associated CLIP-170 initiates herpes simplex virus retrograde transport in primary human cells in the paper by Jovasevic et al., published in the October 26, 2015 issue of the Journal of Cell Biology.
You